I am happy to report to you all that we are running not on Eastern Standard Time, not even on Pacific Standard Time. We are running on people time. We are running on relationship time. Therefore, according to the clock, we're late, but according to reality, we're always on time. And here we are, but we should start. So if you would kindly take your seats in preparation for an earth-shaking presentation by a scientist that nobody dares peer review. I introduced you to David Johnson, who has another stellar quality, and that is he laughs at my jokes.
We all know the existing reality that faces us every day. We have an increasing population, resource conflicts, and migration. We have 30% loss of agricultural land over the last 40 years. We have declining soil fertility on the remaining 70%. We previously had an average of 18 inches of topsoil. Now we have 8. 21 of 37 of the world's largest aquifers are being depleted faster than they can be refilled. And before 1940, it only took 1 calorie of fossil fuel to produce 6 calories of food energy. Now we use 10 calories of fossil fuel to produce 1 food calorie. And thank you, Adam, for this. It's at the bottom of his emails. You never change things by fighting the existing reality. You change something by building a new model that makes the existing model obsolete. So, we probably need to explore the past and see how we're maybe able to shape our future. Biology has shaped this planet for the last 4.2 billion years. It has done this by increasing both efficiency of the capture, storage, and utilization of energy from the sun, and increasing system biodiversity to provide resilience to the system. Microorganisms regulate all of the elements through biogeochemical systems. Take copper. Before we can mine it, or now how we mine it, we use microbes. Aluminum goes through 2 microbial processes before it's economically viable for us to process it. Iron, in the Earth's crust, it's too low of a concentration to really be able to mine it, but microbes in the sea attract iron molecules to the surface of their cell. They get heavy and they settle down to form bands of iron that we can mine profitably. We use almost all the elements on this periodic table in the protein structures of either to build the structure of our cells or as an enzyme, as a cofactor in a protein structure. This is the number of biochemical reactions that go on in your body every second of every day. When looking at the past, water came on the planet about 4.2 billion years ago. Bacteria came along at 4 billion years ago. Cyanobacteria and photosynthesis, the best evolutionary development on this planet that has made this planet what it is today. The ability to capture, to store, and utilize energy at a later time. This led to the great oxygenation event, and if you're an anaerobe, this was not good news. But if you were a fungi, this was good news. That allowed you to come onto the scene. That led also the ozone oxygenation led to the buildup of the ozone layer, the first single-celled animals. Gomorrhamacata and land plants developed about the same time in the fossil record. Now, Gomorrhamacata are the mycorrhizae that associate with a plant to go out and extract nutrients from the soil parent material in exchange from energy products from the plant. We are the new kids on the block. We're up here. We're not even a blip. Looking at the last 600 million years and the atmospheric CO2 concentrations, you can see we've been as high as 7,000 parts per million. Black is your CO2 in the atmosphere. The red, this right-hand column, is the oxygen in the atmosphere. And the blue is the temperature that we've had on this planet over the last 600 million years. We are here. Probably the best time on this planet. 400 parts per million CO2, about 20% oxygen, and temperatures of about 14 degrees C. Let's look and see how life has influenced all these ups and downs of oxygen and CO2 and temperature. In the Cambrian era, we had a Cambrian explosion, corals and shelled animals. They led to a precipitous drop in CO2. Let me add one more thing here. Down here at the bottom are our extinction events. And you can see each extinction event led to an increase in CO2 through this process. Back to what the plants and animals and microbes were doing. The first land plants came in about 500 million years ago. You can see another drop. Each step along this process is an increase in the efficiency of this system to capture energy and store it and pull CO2 out of the air. Lignin and vascular plants developed right about in here. Again, another drop. Then we had the extinction event, a blip up. Also, the coral reefs played into this. Coral reefs have come and gone in the fossil record as they've evolved in and out of the system. At this point, we had 8-meter tall fungi, progymnosperms, roots, leaves, and seeds. This gave plants the ability to colonate all of the planet, not just to be pollinating around where water was. And you can see we had a precipitous drop in CO2 down to almost the levels that we have today. So this is when we had coal seam development. And one of the reasons that we had coal seam development, we did not have lignin-degrading microbes. Well, nature took care of that, brought them in about 275 million years ago. You can see an increase in CO2 from them beginning to degrade the plant material that was growing. At this point, conifers, ginkgos, and evergreens came in. Angiosperms again, or for the first time, coral reefs again. About 55 million years ago, we had a cooling period, landmass positioning changes, but most importantly, we had mammals and ungulates come on the scene, as well grasses did not evolve until this time. So the grasses and the ungulates, these systems of grazers, have what led to the soil carbon buildup that we see today. Microbes are the backbone of every organism and ecosystem on this planet. As you can see in the stomach, the mouth, soil, and the root nodules, microbes are everywhere. You have 10 trillion cells in your body. You have 100 trillion microbes. You have about 30,000 genes in your body. They have 8 million genes. What do they do for you? Just about everything. I kind of see us as an artificial intelligence experiment gone horribly awry. They digest our food, they do cell renewal, they comprise 60% of our immune system, vitamin synthesis, prevents allergies and skin disease, controls your appetites and cravings. They turn on and off genes that regulate brain development, anxiety, depression, arthritis, and emotional behavior. You could also be the cruise ship for these guys because you feed them, you house them, you transport them, and I'm sure you provide entertainment. What happens when these microbial communities are disturbed? Something like Crohn's disease. Many people suffer for decades with this. They usually get it from when they've taken too many antibiotics and they destroyed the microbial community structure in their intestines. They found out that you can do a fecal transplant from a healthy donor and cure people within 24 hours. Some people that have suffered decades with this, and yet they can get a cure within 24 hours just by restoring that microbial community. Well, we've done something similar to this in agriculture. Almost every process we have, the plowing, the application of herbicides, pesticides, all of these have led to the destruction of that soil microbial community. So what if, it's a big what if we could just restore that soil microbial community with an inoculant, a healthy inoculant? And that's kind of the trip I'm going to take you on today. First of all, we need to understand how nature has this system set up. As you can see, in a bare soil parent material, we're talking predominantly bacteria. But as the systems improve, going from bare soil parent material up to old growth forest, your climax ecosystem, you can see you change to about 100 to 1 to 1,000 to 1 fungal to bacterial ratio. So you're predominantly fungal. At the same time, you see increases in productivity through this system. In your deserts, you're only seeing 90 grams of dry biomass per square meter produced every year. And yet, in your old growth forest, you're seeing 2,200. There. This is a bacterial dominant soil. We're in a microscope, we're focusing down through the system. Each of these is a bacteria. You can see by their movement around. And as we focus down, you also start to see the minerals in this. This is what a healthy agricultural soil looks like. You have significant organic matter with fungi attached and growing on it, and you still have the biology of the bacteria. This is what the Muir Redwoods forest looks like, that climax ecosystem. You can see very few bacteria, more fungi. So, where are we today in agriculture? We grow weeds really good. Where we need to be is here, in your late succession of grasses and your row crops. And the way nature has done it, or the way we're going to do this, and the way we've done it in the past, is controlled disturbance. We have to keep this system right in that area, or to 1.1 to 5 to 1, for the crops that we enjoy eating to be able to be grown. This was nature's controlled disturbance. Before the settlers came, the bison, 60 to 80 million bison roamed the northern plains. It was that system that developed the deep soil carbon profiles that we enjoyed for the last 200 years here. They would graze this grass. They'd only take about 40% because they were always being herded by predators. Then the manure they would spread, plus the saliva as well, would spread microbes. Okay? Sure. They don't want to see me.
Oh, yes,
They want to see this.
well, that's prettier, but we're going to see.
Okay. It was a system that developed because you also depended on the insects in this system, the dung beetles, to roll up the dung that they would spread and put it in the ground, and it would basically compost in place. So what we've tried to do is duplicate this system in agriculture by making a compost to replace what the dung beetles do. You know, everybody wants to replace a dung beetle for a job. My wife and I developed a composting process that's static. One of the big problems, we developed this with USDA. They wanted us to do something good with dairy manure. This was the job I got the second day I got to work. You can imagine my elation. Do something good with dairy manure. The problem with dairy manure is it's very saline, very salty. And the people that had this project a decade before me concluded that compost was bad for soils. And I laughed at first, but then I read their data, and they were right. What they were ending up with in their composting process was a compost that was just as saline, if not more saline. You start with about 30 to millisiemens, and they were ending up with 44 millisiemens. Plants don't like anything more than 10, and most can't stand anything more than 3. What this composting process did, it reduced water usage by 6 times in our area. It reduced composting time by about 66%. It can take about a year and a half or more in our area to compost something. It resulted in a low salinity compost, around 2 to 3 millisiemens. It was amenable to incorporation with vermicomposting after the thermophilic phase, and that's an important part. If you do this, you need to have the worms in this process. But most importantly, it gave us a fungal dominant compost with which to inoculate these soils. This is what it looks like through the composting process. You can see the temperatures over here on the left hand. These colored bars represent all of the different bacterial phyla. You can see through the process there's an ebb and flow of these organisms. Each one has a particular job at a particular time, and as you go through the process, you start to break the material down. we looked at a compost made a decade ago, the analysis of the bacterial phyla, to a compost made this year. This compost was made with 1/3 dairy manure, 1/3 yard waste or yard trash, leaves, and 1/3 wood chips. The bottom one was made with all leaves. And yet you can see there's a striking similarity between the structure of the ending microbial community. At 4 weeks, this would be the bacterial species structure of the compost that I do. You can see it has about 23 species in the top 80%. There's not a lot of diversity and there's a lot of unevenness. Some populations are higher than others. This is at 22 weeks. Everybody says this is when compost is done. Well, maybe not. This is at 60 weeks. You have a 4 times increase in the diversity of the system the longer you let it mature. And I think that's one reason when you see the results that we're seeing such impressive growth when you inoculate with this compost. Of course, looking at this, the blue bars here represent plant growth. These 2 on the left are the compost that we used. On the far left is a compost we'd put into the greenhouse 5 months previous to this growth test. We'd watered it daily to flow through just to see if we'd leach these nutrients out, how is this compost going to do? We can see the one on the left, the leached one, did better than the one straight out of the compost pile. We also looked at several inorganic tests, nitrogen, phosphorus, and potassium. I was an NPK junkie. I'm 13 years reformed. You can see many of them had much higher nitrogen content than ours, and yet we had better plant growth. So this kind of struck us as odd. Phosphorus was the same way. Potassium, again, no correlation, plant growth to available nutrient. Thought, organic matter, that's the key to all this. No, only 0.12. We had taken a biological test, a fungal mass to a bacterial mass. It had a pretty good correlation. We weren't expecting this, so this was a side road that we took. When you look at the change in the soil from bacterial dominant to fungal dominant, looking at the partitioning of carbon into, well, the brown is the root carbon, the green is the shoot carbon, the red is the fruit carbon, the gray is new soil carbon over and above what we started with in this test. The blue is respiration carbon out of the soil, and the orange is carbon that was used to fix nitrogen. You know, consider that it's very expensive energetically to fix nitrogen. We do it in the Haber-Bosch process at 500 degrees C, 2000 psi in an iron catalyst. Nature does it at room temperature and standard temperature and pressure. You can watch the efficiency of the system as you move from bacterial dominant to fungal dominant as far as the efficiency of how much that carbon goes to the plant. In a low carbon bacterial dominant soil, we see 3% of that carbon flow actually to the plant. 0.84 on the fungal to bacterial ratio, 17%. As you increase, 21%, 30%, 37%, up to 56%. That's where we are in agriculture. We have bacterial dominant soils with low carbon. We're running about 10 to 11%. According to this, we should be able to quintuple the productivity of these soils if we bring them up to a higher fungal to bacterial ratio and return this carbon to the soil. When you look at the investment by the plant of the carbon that it dedicates to the soil to improve that community, in the bacterial dominant soil, 96% of that carbon flow went to the soil to improve that biology. That's a significant investment in risking the life of that plant to put that much energy into the soil. And yet, even in a very healthy soil, still over 50% of that energy actually goes into the soil. So how well does this work in the real world? To get a clue of how things are, net primary production, we were talking the number of grams of dry biomass per square meter produced per year. That's net primary productivity. In a desert, we're looking at 90. In the tropical rainforest, your best terrestrial ecosystem, about 2,200. Here we are, cultivated land, 600. We water it, we fertilize it, we weed it, and yet all we are averaging out of this is 600 grams of dry biomass. We're doing something wrong. What I'm proposing is a biologically enhanced agricultural management, or BEAM approach to agriculture, where we restore that biology to this system. In my transitional BEAM, I've been able to match the most productive ecosystem in the world on annual productivity. In advanced BEAM, I've seen over 3,000 grams of dry biomass per square meter. BEAM employs inoculating these soils with the compost. We first started out at about 400 pounds per acre. This is what it would look like on 1 square foot. It is a dusting only. You're only putting in microbes on this system. You're not putting in carbon, you're not putting in nutrients. This is a completely different way to look at compost. It's actually, when it's made right, it's an inoculant. This is our first year of BEAM application on the right, and the control is a conventional approach. This is on land with most likely legacy issues. It's at the Plant Science Farm at New Mexico State University. They've done probably everything to this soil. But as you can see, after these were 1 year BEAM first, and then these fields were planted at the same time with the same crop and watered at the same time. We saw a 5 times increase from 50 grams per square meter to 250 grams per square meter after the first year. Year 7, we're now seeing 1,141 grams. When you go back and look, 250 grams per square meter after our first year, we've almost quintupled the productivity of this soil, just with adding 1 time compost addition. You can also see there's 387 kilograms of nitrogen in this above ground biomass that we fixed and put into the system. Nitrogen is the problem.
Okay, clarification?
Yes.
You're saying that this was a one-time application 7 years prior
Yes.
and none since?
The rest of the process, it's about 2% inoculation and 98% management. You need to have something growing on there all the time in order to get that carbon flow in to support the microbial community. Yes, ma'am?
So are you saying it wasn't tilled at all during that period?
I disc. I don't till. I don't turn plow.
Just chew a little bit?
In this, I had 2 experiments that I set up. I had one where I'm putting everything back because I want to see farmers to be able to use this as a carbon market. So if they can dedicate their land to farming carbon, they can make a profit on it and improve their soils at the same time. I also have another test that I implemented in a desert soil, a non-legacy soil, where I extracted everything. I'm trying to find the both ends of this paradigm, so we find out somewhere in the middle. We're going to have to give a certain amount back to the system in order for it to survive. We can't extract everything, as you'll see here, but it's having the 2 bookends to see where in the middle that it does the best. This was a summer cover crop just before that. It's a mixture of black oilseed sunflower, Sesbania, and a forage soybean, and a millet. June 23rd, it was about 4 to 6 inches tall. 29 days later, it was about 6 feet tall. 17 days later, it was over 7 feet tall. Now, black oilseed sunflower is only supposed to get 4 to 5 feet tall, only supposed to have heads about this size. We do not know what a plant can do with the right biology in the soil. We've spent 75 years trying to change plants to grow in a poor soil, when indeed we could have probably thought about changing the soil. This is Sesbania. The first season I grew it was 4 feet tall. The second season was 6 feet tall. The third season was 12 feet tall. These are the dynamics and the change of this soil as you build this microbial community back up. They offer a lot of potential for agriculture, I think. This is a desert plot. You can see where I live. Very green, lots of trees. The first season on this, and again I did this so a non-legacy soil, because I know there's issues in agricultural fields that will affect the productivity until we get the biology to clean it up. The first season, The first winter cover crop I grew was 800 grams of dry biomass. This is the second one. That's 1,608 grams. And look at that, 486 pounds of nitrogen that we fixed in that above ground biomass. 2,190 grams of dry biomass in the third crop that we grew. 654 kilograms of nitrogen. Now, this is a winter cover crop. We're planted in October. It doesn't really wake up till mid-February. We take it out of the field, or this is the one I take out. We take it out at the end of April. This is 10 weeks of plant growth, and we've equaled the most productive ecosystem in the world on productivity. That's how much potential we see. This is the soil I started with. There's no A horizon. It's just sand. After 3.5 years in the control, where I just grew the cover crop, cut it and hauled it off. Sorry. And in this one, we had inoculated the first year and did the same thing, grew cover crops, cut them and hauled them off, and planted again. There's over twice the depth of the A horizon increase between a compost treated and an untreated. This is what it looked like over the first 4 seasons of growing the winter covers. In the regular soil, we got 492 grams of dry biomass. In the finish, 2244. So we had a 5 times increase in that productivity there. You can see here, the yellow is the control, the green is the compost treated. We're at 30, 31, almost 3,200 grams of dry biomass per square meter in the year. In that, we also saw 872 kilograms of nitrogen per hectare per year fixation in the above ground biomass. That's about, if you want acres, it's 777 pounds. Most crops won't take more than 180 to 200 units of nitrogen. You can see you have adequate nitrogen in this system. When we look at a metagenomic analysis of these 3 soils, this is my desert soil, the control, and the compost treated. This is a heat map showing the percentage of population of that particular either phyla in bacteria or eukaryota, where your fungi are, or archaea. You can see by the heat map, the compost treated soil has much more diversity. If we take one of these, where you know it's dark to light to nothing, proteobacteria, that's a really good community. In proteobacteria, we have gamma proteobacteria. In that phyla structure or substructure, we have free-living nitrogen-fixing bacteria. We have phosphorus solubilizing bacteria. We have bacteria that secrete plant growth hormones. We have 21 other categories in this that we could explore to depth that show the same indication in that heat map of higher concentration to low. Diversity is the currency of survival, and that's what's making this system work so well. So what we're showing is increases in carbon captured in the soils. Over the first 4.5 years of that the land decker treatment, we were just putting everything back, trying to see how fast we could build carbon up. We're averaging a 0.24% increase in the top 30 centimeters per year. That's 10.7 metric tons per year carbon per hectare per year, or 37 metric tons of CO2. In 67 long-term no-till studies by West, the carbon capture rate was only 0.5 ton, 0.57 tons of carbon per hectare per year. Nigley, with agri-ecosystems where they've mulched, was only looking at 0.2 tons carbon per hectare per year. And Lowell has seen the possibility of 0.7. The first 4.5 years, we've averaged 10.7 tons of carbon per hectare per year increase. And we see the potential up to 19.2 in a system that's already improved. At this rate of carbon capture, it would take 50% of arable land to capture all anthropogenic CO2. At this rate, it would take 25% of arable land to capture all anthropogenic CO2. You realize we're only looking at a 0.25% carbon increase per year in our transitional and 0.5% of increase soil carbon increase per year in the theoretical. I felt a little bit out on the limb till Gabe Brown came along. Thank you, Gabe. Yes, let's give Gabe a hand. The first 13 years, Gabe tried no-till, he tried crop diversity, he tried cover crops. He was only averaging 0.1% increase per year. Once he got to 3% soil organic matter, and that is, I'm seeing that as a key in this, and a lot of farmers see 3% soil organic matter, they see a change in the dynamics of how their soil works. So that's also part of it, the amount of carbon. But once he got to that, once he stopped using nitrogen fertilizers, started implementing multi-species cover crops and livestock integration, he changed that slope to over 0.5% per year, better than what my model predicted. So we're also seeing an increase in soil carbon use efficiency. We can put all the carbon we want in the soil, but if it does not stay, it's not going to mean anything. Most scientists say the more carbon you put in the soil, you're going to have a linear response, you're going to have respiration, and it's just going to respire out. Now, there is an increase in carbon respiration as you go through this system. This is the bacterial dominant, this is the fungal dominant. But you have to look at this system entirely. There's an 18 times increase in beginning soil carbon in this test. There's a 5 times increase in microbial biomass, but only a 4 times increase in respiration. When you look at this a little different way, looking at the percent of the initial carbon in that soil that you respired, in a bacterial dominant soil, we're seeing over 50% of the original carbon in that soil respired. But in a healthy, high carbon, high fungal to bacterial ratio, only 11% is respired. Nature uses this system. She's used it. I mean, there's no way nature could have accumulated as much carbon as she had in the soils without this system functioning. Because we had, again, the 18 times increase in soil carbon mass and a 4 times decrease in relative soil carbon respiration. This system also increases the availability of macro and micronutrients. This is a big issue as far as the quality of the food that we're getting off of the soils. Our addition of nitrogen and phosphorus and potassium have outstripped the ability of these soil systems, the microbial community, to extract the micronutrients out of the soil. In a 20-month study, when I first started initiating beam on these soils, 5 sampling periods, I saw 1,100% increase in both manganese and iron in their soluble water plant available form. I saw an 83% increase in magnesium, 76% in calcium, 60% in zinc and copper. I also saw increases in P and K. And this is a system I've never put fertilizers in. I've never put anything into this system. It's only the biology that is actually able to release these elements from the soil structure. We're also able to increase storage and availability of water. In our area, the first 1% increase in soil carbon quintuples the water storage capacity of that soil. We've got nothing to lose by starting to put carbon back in these soils. It also improves crop yields. This is James Barrowfield, Los Alamos National Laboratories. He's the one that works on the Space Vehicle Curiosity on Mars. He's the one that developed the laser-induced breakdown spectroscopy. We were having him out to be able to try to get a quick and dirty test of the carbon increase in our soils. We just happened to be out when the cotton was getting somewhere near ready. In our conventional cotton with 180 pounds of nitrogen per acre, compared to a beam transition after 1.5 years, we had over double the productivity without fertilizers. Now, cotton is the dirtiest crop in the world. It takes more fertilizer, more herbicides, more pesticides than any crop on this planet. When we did a preliminary test, we could see using beam, we could have over a doubling of cotton production, conventional cotton production in our area, and a 98% increase in chili. That's another one that grows well in our country. Now, conventional organic takes a big hit on productivity because it's basically a conventional approach using natural amendments or fertilizers. They're still in the amendment phase instead of regenerating the soil and having the soil microbial community do that extraction process for you. This was July 17th this year on our cotton. This is the second, about 2 weeks later. This is August 29th. High cotton doesn't mean anything unless you've got bolls on there. And bolls were so heavy on this, the plants were starting to droop down. And this is what it looks like now. I haven't harvested it yet, but it's a pretty good yield for no fertilizers, no herbicides, and no pesticides. So I think we can meet the challenges that we have ahead. We can reclaim the degraded soils and increase land area for agriculture, or we might be able to use less land area if we can grow twice the crop on the same amount of land. We can produce more food in local soils. That means Africa, that means the Middle East, that means South America. We can use less water to grow crops by increasing both the plant water use efficiency in this system and the soil water storage capacity. We can also reduce energy use by allowing nature to extract and fix all the essential nutrients those plants need. We can reduce both agrochemical pollution and atmospheric CO2 at the same time. Why should we pursue this? There is no planet B.